We have just reviewed the genome architecture of the coronavirus family, a single-stranded positive sense RNA, some 30,000 bases long, that corresponds to two large polyproteins, followed by a nest of subgenomic segments that code for the viral structural proteins, spike, envelope, membrane, and nucleocapsid proteins. As it turns out, coronaviruses are pretty common. They all have RNA genomes following the same general layout, and based on their similarities or differences in RNA sequences, they can be clustered into four genera: alpha, beta, gamma, and delta coronaviruses, forming the coronavirus family tree, or phylogenetic tree. The science of phylogeny traces the historical pattern and relationships amongst organisms reflecting their evolution. Organisms that share common traits cluster together on the same branch of the tree, which makes phylogeny so important and powerful. When we discover a new organism, we can place it on one tree limb or another based on RNA genome sequence. This immediately provides insights about its biology and what to expect about this new member based on what we know about its closest relatives. Thus, for example, alpha and beta coronaviruses infect mammals ranging from mice to man, bats to beluga whales. Not surprising is that 229E and OC43 can be placed with alpha and beta coronaviruses. The gamma coronaviruses are exclusive to birds, and the delta coronaviruses primarily target birds as well. Let's look first at coronaviruses that infect our pets. Cats are often infected by feline coronavirus, a member of the alpha coronaviruses that typically infects cats' intestines, causing a mild enteric disease which becomes chronic, asymptomatic, yet still highly contagious, indicating that immunity may not be long-lasting or highly effective in clearing the infection or preventing recurrent infection. The virus can mutate, generating a variant virus, feline infectious peritonitis virus, that shifts to target and infect macrophages. This tropism switch leads to a severe disease affecting the liver, lungs, and brain, producing a cytokine-driven inflammatory fatal disease. Preventive vaccines are found to be only partially effective and require annual boosts. Dogs also suffer from alpha coronaviruses. Canine coronavirus was discovered in 1971. This virus causes damage to the lining of the intestines, leading to vomiting and diarrhea. A killed virus vaccine has been available since the 80s, and although it does reduce serious disease, it does not afford protection from recurrent infection and subsequent transmission. Coronaviruses cause common infections in many domestic livestock, causing substantial commercial burden. Pigs, for example, suffer from at least 6 different coronaviruses, like the alpha coronaviruses of cats and dogs. Porcine endemic diarrhea virus causes an enteric disease that older hogs recover from. However, newborn piglets are especially vulnerable to PEDV and usually die from a severe disease within days of getting infected. Over the last decade, a number of vaccines have been developed primarily for vaccination of pregnant sows, conferring some protection to suckling newborns. Here too, however, the effectiveness of these coronavirus vaccines is only partial, reducing fatalities by 40 to 50% and requiring multiple vaccinations over time. One rather intriguing swine coronavirus is transmissible gastroenteritis virus, another alpha coronavirus, that similarly is age-dependently fatal for newborn piglets. Here, nature has introduced an unexpected twist to the story. A genetic variant of TGEV has emerged that targets the lungs, causing a mild, manageable respiratory disease. This tropism switch apparently is the result of a 224 amino acid deletion in the viral spike protein. The mutant variant, called porcine respiratory coronavirus, although highly contagious, is much less dangerous and has led to effective herd immunity, not only to itself, but against the progenitor TGEV as well. Thus, PRCV in essence provides live attenuated protection against the fatal enteric piglet disease caused by TGEV. Coronaviruses infect cattle as well. Bovine coronavirus is a beta coronavirus that causes both enteric and respiratory disease in cows and calves. Although infection leads to relatively high antibody concentrations, natural protection wanes over time, and reinfection often occurs. The vaccines that have been produced require multiple boosts in order to provide effective protection. Gamma coronaviruses of birds, such as avian infectious bronchitis virus, cause respiratory and kidney disease in chickens, affecting egg laying as well. Although a number of vaccines have been developed, protection is often short-lived and waning with only a few months. So, what have we learned? Reviewing the diversity of the viruses amongst viral genera, the diseases they cause in mammals and birds, the natural immune response, and age-dependent mortality, along with variable protection by vaccines, seem to reveal a pattern. Coronaviruses typically cause enteric and respiratory diseases. Natural infection leads to what seems like only transient immunity and consequently recurrent infections. Age dependence is associated with severity of disease outcome. Emergence of genetic variants can lead to more infectious, highly transmissible viruses with either milder or more severe disease presentation. Vaccination is possible, although vaccines seem to reduce severe disease, but not necessarily the ability to block infection or transmission, and continuous protection requires multiple and repeated boosts for best results. Sounds familiar? Let's now move on to the coronaviruses that infect people.